So in this topic we describe development of male and female reproductive system. So the competency is development of male and female reproductive system. The genetic sex of an embryo is determined at fertilization by the sperm that fertilizes the oocyte. Gonads do not acquire male or female morphologic characteristics until the week 7 of development. Primitive genital system. Gonads, that is testes and ovaries, are formed from two types of cells: reproductive germinal cells or primordial germ cells, nutrient supporting cells, that is follicular cells of the ovary, Sertoli cells of the testis. Primordial germ cells are discernible at about 21 days of embryonic life, are seen among the endodermal cells in the wall of the yolk sac near the origin of allantois. Primordial germ cells migrate by amoeboid movement along the dorsal mesentery of hindgut during week 5 and reach lumbar region of the developing embryo that is going to form the future gonadal ridge. Coelomic epithelium lining the mesonephric body thickens to form the genital or gonadal ridge and provides nutrient supporting cells of the gonad. If the primordial germ cells fail to reach the ridges, the gonads do not develop. In week 6, the primordial germ cells invade the genital ridges and are incorporated into the primary sex cords, which proliferate and grow from coelomic epithelium into the underlying mesenchyme to form the primary sex cords. Gonad is indifferent or undifferentiated at this stage because it has the same morphologic appearance in both male and female. Indifferent gonads consist of an outer cortex and an inner medulla. In embryos with XX sex chromosome complex, the cortex forms an ovary and the medulla regresses. Embryo with an XY chromosome complex, the medulla differentiates into testis and the cortex regresses. Sex cords become seminiferous tubules in the male. Primary sex cords continue to proliferate and anastomose deep in the mesenchyme and produce a complex network called the rete testis. Rete testis, with adjacent part of the mesonephric tubules, establishing the initial and first urogenital connections. Towards the end of the month 2, the mesonephric body begins to regress, the glomeruli disappear, and only the mesonephric tubules remain linked with the genital gland. Male gonads develop into testes towards week 7 of development. The differentiation is determined by the XY genetic constitution and proceeds. Primary sex cords proliferating from coelomic epithelium condense, extend into the medulla of the gonad. In the medulla, the cords branch, they deepen in anastomose and form the rete testis. Sex cords become seminiferous tubules or testicular cords. They lose connections with germinal epithelium because of the development of the thick fibrous capsule, they lose connection with germinal epithelium because of the development of thick fibrous capsule, that is tunica albuginea. Development of tunica albuginea is the characteristic and diagnostic feature of testicular development. Testicular cords develop into seminiferous tubules. Deeper portions narrow to form straight tubule, converge on the rete testis. Seminiferous tubules become separated by mesenchyme, which gives rise to interstitial cells of Leydig. Androgenic hormones secreted help in the differentiation of the genital tract and the external genital organs. Interstitial cells of Leydig reach their maximum development between 3.5 to 4 months. Walls of seminiferous tubules, as a result of their cellular duality of origin, are composed of two types of cells. Supporting cells are sustenancular cells of Sertoli, derived from the germinal epithelium, and spermatogonia derived from the primordial germ cells. Germinal epithelium flattens to form the mesothelium on the surface of the testis. Rete testis become continuous with 15 to 20 adjacent persistent mesonephric tubules. Persistent mesonephric tubules participate in the formation of excretory tracts of the testis. Efferent ductules open into an adjacent mesonephric tubule, which becomes the ductus epididymis or epididymis. Thus, vasa differentia and the epididymis are of mesonephric origin. Thank you.